Welcome to Life in Biology. I'm Dr. Joel Graff, and today's video is going to be about some molecular biology basics, but once you understand it, it'll help you build a foundation for new information about molecular biology. And the foundation, in my opinion, of molecular biology is the central dogma of molecular biology. Now this shows a somewhat stripped down version of the central dogma of biology. I'll have some more charts here in a second that have more arrows to them. But in the central dogma of biology, one of the important points to take home is that there is a flow of information, genetic information, from DNA through RNA and to protein. And the process of reading DNA and creating a new strand of RNA is called transcription. So DNA and RNA are both nucleic acids, so that's the molecular or macromolecule class that they fall into. And so if you go from one language to the same language but a slightly different form, it could be transcription, like a transcriptionist would listen to something in English and write down those words in English. When you go from RNA to protein, that process is known as translation. And as you would translate languages, it means that you go from one language to the next. So RNA being read and made into a new language of protein would be translation. So that's the general flow of information, but we're not quite up to the full central dogma of microbiology yet. Instead, we'll see those shown here. And with the central dogma of molecular biology, as you go from one type of molecule, DNA to RNA to protein, as you go from one molecule to the next type, you will need an enzyme to read and make a new copy of things. And so there's a lot more arrows on this particular slide, and what I'm pointing out here is that here we've got DNA going over to RNA, and that's going to be carried out by a polymerase, which is what the small p stands for here. But I have DDRP written. What that means is it's a DNA-dependent RNA polymerase, which means it reads DNA and it makes RNA. Okay? When you go from RNA to protein, the enzyme is called a ribosome. But there are other steps where you can have additional polymerases come into play, and not just that DNA to RNA like the previous slide showed. And what we're going to see here is that if you have some DNA, your cell might want to copy that DNA to make a new cell. So you have to copy your genetic material. In that case, you would use a DNA-dependent DNA polymerase. Reads DNA, makes DNA. Some viruses and some other cell types, some viruses encode this next enzyme, but it's also found in other cell types, other animal eukaryotic kingdoms such as plants and insects. And this polymerase allows the cell to read RNA and go back towards DNA. So it's the reverse of that process we called the reverse of the process we called transcription. So going from RNA back to DNA is known as reverse transcription. So again, how you describe that polymerase is that it's RNA-dependent DNA polymerase. It reads RNA and then it can make a new copy or a new strand of DNA. And some RNA can be copied. And so if you start with RNA and you make a new copy of RNA, the enzyme that would do that would be an RNA-dependent RNA polymerase. So there's different steps or different enzymes that carry out these whole things. One thing you'll notice is that although you can replicate DNA with DNA, you can replicate RNA with RNA, and you can go from DNA to RNA and RNA back to DNA, one thing that you can't do is you can't go from protein back to RNA. and you can't go from protein back to DNA. And when Francis Crick came up with this concept of the Central Dogma of Biology back in the 1950s, the main point of his paper was to say that you cannot take something that's in the language of protein and get it back into the language of DNA or RNA. And the reason for that is that it takes 3 pieces of information of RNA to make a 1 piece of information about your protein. And we'll get into it later, but you can't take that 1 piece of information about protein and get back to always the same information about your RNA or your DNA. So there's no backwards from protein. So protein's kind of the end product in this line. And so we're going to be talking about proteins a lot. The last concept I want to bring up with the Central Dogma of Biology is the idea that each of these steps that we've talked about can be regulated. When you have DNA and you want to copy it to make more DNA, that needs to be regulated because you don't want cells to replicate out of control. And so you can control the speed at which DNA gets made into new DNA, or the timing of when that process begins and follows through. Also, as you go from DNA to RNA, I have a large arrow and a small arrow to indicate that the rate of reading DNA and making RNA changes. So in your DNA, in human cells anyway, there are 20,000 genes. And in those 20,000 genes, they can individually be regulated how fast they are being made into RNA. And so a lot of molecular biology is following the regulation steps of getting from DNA to RNA. RNA can be translated to protein. That was the ribosome, if you remember. And the efficiency at which RNA can be read and made into protein can also be regulated. So this is called PostTranscriptional gene regulation. So the rate at which RNA gets made into protein. And the final piece that I want to talk about is that these molecules can be degraded. DNA will be degraded if a cell decides to go through programmed cell death, but all-out degradation of DNA is pretty rare. Each RNA molecule, however, that's made has a certain half-life, how long it'll stick around in the cell on average. And sometimes that half-life can be very short, so you'd have a large arrow going down to degraded RNA, or the RNA could be stable, and so the arrow leading to degraded RNA might be somewhat a slow process. Same thing with protein. Protein can be degraded down, can become degraded protein, and the rate at which you degrade protein can also be regulated. And so when cells are trying to control which proteins, that's the end product of this flow of genetic information or gene expression, which proteins are going to be available in the cell, there are a number of checkpoints and steps in the pathway that can be regulated. And so these different steps are going to come back time and again as we learn more and more about molecular biology. All right, have a good day.